for the production of protein. All right? Now, these two processes, from DNA to RNA, all right, that occurs in the nucleus. And since we're using the language, so to speak, of nucleic acids, this process is called transcription. Okay? To transcribe means to rewrite. To write over and over and over again in the same language, essentially. Okay? To transcribe. Whereas when we go from RNA to protein, since protein consists of amino acids, we have a whole new language. So this process, which takes place here in the cytoplasm with the assistance of the ribosomes, that process is then called translation. All right? And I'll describe translation in more detail in a later video when we get to Module 4 on heredity. It'll be important then. Okay, so as now the transcript is read by the ribosomes, it moves down messenger RNA, a particular sequence of amino acids according to the gene, all right, according to the sequence of ATCs and Gs, is produced. And as this occurs, the protein produces its three-dimensional conformational shape, and most proteins are globular. So here I'm describing this now. These proteins are forming here in the cytosol, okay, as the messenger is being read. All right, I hope you get that basic idea there, okay? All right. Remember, there's tens of thousands of different proteins, almost all of which are enzymes floating around here in the cytosol. Okay, I'm going to erase this. Make sure you understand this process, DNA to RNA to protein. I'm going to be using the words transcribe and translate again and again. All right, know the essential differences between DNA and RNA as well. Okay, now, if the message should attach itself here to one of the ribosomes, one of the attached ribosomes to the endoplasmic reticulum, and in which case, of course, we call this rough ER because it just looks rougher in electron microscope pictures, electron micrographs, a rough ER. Then the protein product is imported through tiny holes. It threads its way through tiny pores in the endoplasmic reticulum. Remember, the membranes here, all of these membranes are all bilipid layers, okay, composed of phospholipids and protein, about 50-50, although the composition of membranes is different. So the composition of the proteins and their associated enzymes found in endoplasmic reticulum would be different than those found in the Golgi apparatus. All right, so as translation occurs and these protein products are produced here in the Golgi apparatus, they're modified, usually by the addition of sugars, and I'm showing that here in green. Eventually, vesicles are formed, membranes at the terminal end here, okay, will form with those protein products that have been glycosylated. All right. Okay, so I show here now a vesicle pinching off or budding off, and then moving towards the Golgi apparatus, where it will fuse here like that. Okay. So when it comes into contact with the membrane of the Golgi apparatus, those membranes will will coalesce and fuse together, and the protein will be delivered here. Now, so best to think of the ribosomes as tiny molecular machines that are constantly and furiously producing protein. Remember, protein is the stuff that builds your body and forms the enzymes necessary for all metabolism, all chemical activity that takes place in the cell. Your hair is composed of protein, a protein called keratin, which is rich in cysteine and methionine, two sulfur-containing amino acids. That's why it smells horrible when it burns, okay? And you're coated with this epidermis, which is just essentially dead cells that are sloughing off, by the millions every day. The protein that makes up the lens of your eye, the protein that forms the ligaments and tendons that give your body structure, the protein, the actin and myosin that make up muscle fibers. So protein is essential for structure. Okay, no protein, essentially no life, as far as we know. Now, in the Golgi apparatus, and there might be dozens of them, and especially active cells that are secreting a lot of substances. Goblet cells that line the gastrointestinal tract, for example, are chock full of the Golgi apparatus. They're called dictyosomes, by the way, in plants. All right, and so these vesicles are once again modified in the Golgi apparatus. The Golgi apparatus is sort of like a policeman directing traffic, telling these packages where to go. And so once they're modified again, these substances may find their way to the cell membrane. They may form membranes of other Golgi apparatus. The lipids and proteins that are formed in the endoplasmic reticulum may in fact extend the endoplasmic reticulum. Or lysosomes can be formed. Lysosomes contain a host of digestive enzymes that operate at a pH of 5, and that helps protect the cytoplasm, incidentally. If a lysosome should break open, the cytosol is essentially neutral, so those enzymes aren't very active. But here I show, the process of phagocytosis, where a cell is surrounding some material that it's going to take in. Cells, by the way, are always drinking. That's penocytosis, and phagocytosing, or taking in larger bits of material by this process. Here I show a food vacuole. Now, a lysosome, which was formed here at the Golgi apparatus, actually it would probably be formed over here in this area because there's some processing that goes in that direction. So here we go. Here's a lysosome, with its digestive enzymes. These two membranes can fuse together, and now the enzymes here can break down those food molecules that were taken in by the cell. All right, so I hope that gives you an example of the sort of trafficking and the constant activity that's going on in a